Okay. Uh, so, uh, in last week, what we spoke about in this chapter, okay, we spoke about what are the various, uh, you know, nucleotide derivatives, or you can say the coenzymes or energy storage molecules. We started with ATP. We started with that ATP is the energy storage molecule, right? And then we started we did the NAD, right? NAD positive, which is the coenzyme, right? We discussed that how important coenzymes are, and we did the structure of the NAD positive, which is a dinucleotide. Okay. And ATP is a ribonucleotide. You should be knowing about it, right? Because in ATP we have the ribose sugar. I explained that in the previous class. We have a ribose sugar in the ATP. Okay. While as the NAD is the dinucleotide. Okay. So we finished ATP and the NAD last week. Okay. And today we'll talk about the FAD, which is again which is again a replica of your NAD positive. Okay, it is also a natural oxidizing agent like your NAD. Okay, so FAD is also a natural or a biological oxidizing agent. Okay, and again it is a dinucleotide. Okay, and again it is a coenzyme like your NAD. And I don't need to explain you what is a coenzyme. You know that we spoke about enzymes. We have a hollow enzyme which is made up of an apoenzyme and the non-protein part. Apoenzyme is a protein part, and non-protein part could either be a coenzyme or a cofactor. If it's an organic part, then it's the coenzyme like your vitamins and all. And if it has any element like magnesium, calcium, then it's a cofactor, right? So coenzymes are organic in nature, isn't it? Especially they are vitamin derivatives. Okay, and so is your FAD. Okay, so FAD, as I said, number one, it's a biological oxidizing agent. Number two, it's a dinucleotide. Number three, it's a coenzyme. Number four, what is it used for? It's used for transferring of the electrons, right? So we'll be talking about today about the oxidation reduction reactions, right? And NAD and FAD are excellent agents used for transferring the electrons. Okay, so that is about the FAD. Now let's start discussing about the structure of the FAD, right? As I said that it's a cofactor, right? Let me get my pen. Okay, so this FAD has basically a flavin mononucleotide. As you can see here, this is a flavin mononucleotide present over here. Okay, and then it also has a sugar. Okay, what you need to remember here is that the sugar present in the FAD is little different than what we have in the previously. Okay, so here the sugar is the alditol. So this is called as an ribitol, right? So this is not the normal ribose or a deoxyribose sugar which we have been previously talking about, right? So sugar here is the ribitol. So this is a sugar part over here, right? So flavin is a mononucleotide over here, right? And then as we can see here that this is a base. Okay, mononucleotide base over here. So this is a sugar. As I said, it's a dinucleotide. So every nucleotide should have a sugar base and the phosphate, isn't it? So this is the phosphate part of it. Okay. So the point to remember here is that we don't have a sugar over here, which is a cyclic sugar. We don't have a ribose or a deoxyribose here, the way we spoke about in the ATP or in the NAD. Here the sugar is different, which is an alditol. Okay, so it is the ribit. So there's no cyclic sugar here. Okay, so remember this part. Okay, if you remember this, we spoke about ribitols, alditols, glucitols, sorbitols, right? We spoke about them, previously. Okay, in the carbohydrates chapter. Okay, so other than that, okay, and other than that, what all you need to know here is that, okay, I told you that it contains a base called as the flavin. Okay, and now let's go back to the what is the function of the FAD? Okay, so FAD is basically, when we say riboflavin, first of all, riboflavin is basically the vitamin B12. So the vitamin part of this is, of this coenzyme is the riboflavin. Okay, and which is called the vitamin B12. Okay, and now when we talk about what is the function of the FAD, okay, the FAD is, if you remember, what was the function of the NAD positive? Anybody? So this is it's the same thing what we discussed in the NAD. What was the function of the NAD positive? What do we use as an NAD positive? NAD positive, if you remember, we said that it is an oxidizing agent, right? This NAD positive, once it gets oxidized, right, it forms its corresponding what, reducing agent, and it forms the NADH, which you will see in the once we discuss the various cycles, and you will see how this NAD positive is converted into NADH, right? So same thing is with the FAD as well, right? So look at this, FAD will get converted into its corresponding but FADH2, because as you can see here, one hydrogen is added over here and one hydrogen added over here. Look at this. This is the point where the hydrogen is being added. So that is why it is FADH2. While as NAD positive was NADH, only one hydrogen is added there. Okay, so this FAD will accept the hydrogen, okay, and this will happen during the oxidation process, which we will discuss, okay, in the upcoming lecture today. Okay, and thus this FAD will further get reduced and it will form the FADH2. And this is a very important point. You have to remember, it's the nitrogen, right? It's the nitrogen part of its flavin moiety. This is the flavin moiety. This is the nitrogen, right? So it's the flavin moiety which is active part of this FAD, because hydrogen is added to the flavin part of it. And where to the flavin? To the nitrogen of the flavin part of it. Is it clear? Okay, and you will see that as we will move towards the the upcoming cycles, you will see that how this FADH2 will be converted, will be used to generate the ATP, that we will learn in today's lecture as well. Okay, so that was about the FAD. So FAD and NAD, as I said, that both of them are dinucleotide, both of them are coenzymes, both of them have vitamin part, right? So if you remember, in the NAD we had the nicotinamide part, and in the FAD we have the riboflavin part, or the vitamin B2, because remember, it is a coenzyme, so it should have an organic part, and usually the vitamins act as the organic part for these coenzymes. That is all you need to know about it. Okay, the structure and which part of it is active. It's the flavin part which is active. And remember, what is it composed of? Okay, remember that here the sugar is totally different than the other NAD which we spoke about, or the ATP which we spoke about. Here the sugar is the ribitol, which is a non-cyclic sugar. Okay, and I don't know whether you remember it or not. Okay, if you remember the Hayward structure in the carbohydrates, if you remember, in the Hayward structure, we should have a carbonyl group over here, right? So there's no carbonyl group here. So since there is no carbonyl group, so that's why it cannot form the Hayward structure. Okay. So that's why neither it's a ribose nor it is a deoxyribose. So this thing you have to remember about it. Okay. So the question says, for example, name, for example, just name an oxidizing agent wherein the sugar is a ribitol. Name an oxidizing agent where the sugar is a non-cyclic form, right? So at that time, you should be knowing that it is the FAD, not the NAD. Okay. Do we have any questions about it till now? Are we okay about it? Any questions about FAD? Okay, now we move on to the coenzyme A, right? Coenzyme A is an acetyl carrying group over here. Okay, and it's again a coenzyme, of course. It's a coenzyme A, it's again a coenzyme. Okay, and we know that it's going to help enzymes to function, right? We already know about that. Now it transfers the acetyl group. That is the specialty of the coenzyme A. It helps in transferring the acetyl group. Now, what you need to remember about the coenzyme is that what is it made up of? What is its composition? Okay. But before we start its composition, we first of all try to understand what kind of derivative is it. It is again a nucleotide derivative, right? Whether we talk about ATP, NAD positive, FAD, coenzyme, they're all nucleotide derivatives. So you all can expect that they will have a sugar, they will have a phosphate, they will have a base. Okay, so this is also no different from the others. It is also the nucleotide derivative. Okay, but here the functional group, what is the functional group here? The functional group over here is a thiol group or a thioester group. That is so special about it, right? So thiol group is this is the active part, right? If you see in the last example FAD, we spoke about it's the nitrogen where the hydrogen is added, if you remember here, right? It's a nitrogen with hydrogen. So this is the active part of it. Now, what is the active part of the coenzyme? What makes it so special? It is special because coenzyme A has a functional group which is a thiol group. Okay. Now, this thiol group or SH group, okay, it plays an important role in all our biochemistry reactions. Why is it so? Because of its ability to form the thioesters. Okay, so you don't need to remember that they form the thioesters. All you need to remember is what is the active part of the coenzyme, what makes it special. Okay, the speciality of the coenzyme is that it has a thiol group or the sulfhydryl group, right? So this is this is what makes coenzyme special, right? And it is this sulfhydryl group, okay, which kind of which attaches itself to various other groups, okay, or other molecules can attach to this coenzyme at this part, okay. The way, for example, okay, if you're wearing an abaya, you reach home, what do? You put the abaya in the hanger or in the peg, right, which is attached to the wall, right? So the moment you put the way you put you hang your clothes or your stuff or your abaya, right, to a hanger or to a peg, right? Similarly, this group is an anchoring point, right? So this group can hang all kinds of molecules onto it, right? So that is why I said that this is the specialty about the coenzyme A, because whatever groups have to be attached, right, they will attach on because of the presence of this group. So this group makes your coenzyme A special. Did you understand the activity part of this coenzyme A? What makes it special? Why do we use it to coenzyme A? Sometimes you will see that certain books don't write it as a coenzyme A. You'll see that they will write it as a coenzyme A, and then they will put a word SH here, right? This thiol group over here, right? So why do they put SH here? To show that what is the active part of the coenzyme A. It is the SH which makes this group special. Okay, it is this thiol group which helps in the transferring of acetyl group to the coenzyme A. Are we okay about it? Did you understand why do we use it as a coenzyme A? Because as I said that you should know the reason why are we using ATP, why are we using NAD positive, why are we using FAD, right? What is so, or why are we using coenzyme, right? What is so special about it, about these? Yes, Tanya, right? All these slides are there in the Moodle, but it's that I always add something or underline something or add a video in my slides. Then once we finish the chapter, I upload the slides on the Moodle, right? As a revised PPT. That's what we have been doing right from the beginning. So then it's the students' wish whether they want to study from the PPT which I uploaded earlier or the PPT which I uploaded as a revised PPT, right? We have been doing this since the beginning of the semester, Tanya, right? So once we finish the chapter today, I will upload it there as a revised PPT for chapter this. Okay. Okay, so that was about the active part of the coenzyme A. So what do we use coenzyme A? Coenzyme A is used to transfer the acetyl groups. You will see that in the upcoming reactions that we will use this coenzyme A to transfer the acetyl groups, and that acetyl group is going to get attached over here. As I told you, this is the hanging point. Remember, this is the point where everything gets attached to it. Okay, so we can say that coenzyme A is an acetyl transporting molecule. Okay, so what do you need to know? You need to know that coenzyme A is a coenzyme. Number one, it helps the enzymes to function because it's a coenzyme. It's an okay. Number two, what is it known for? It's known for because it has this, special group over here, which is called as the sulfhydryl group or a thiol group over here. Okay, it might contain one thiol group or sometimes more than that, right? So whenever something has to bind to the coenzyme A, it's attached to the thiol group over here, right? And then we said that the coenzyme A is also called as coenzyme ASH, okay, to show us that it contains this thiol group. And then we said that it is used as what? It's used an agent wherein it can help us in transferring the acetyl groups. So simply remember that it is an acetyl transporting molecule, right? Now look at its structure now. Okay, so I will explain you its structure with respect to the, let me erase all this. Okay, it has a tail, it has a body, it has a head, right? So it has a head, body, and tail. Okay, so tail is this, I call this point as a, that's how I explain students to memorize this structure. It has a tail part. Okay, what is the tail part? Tail part is the mercaptoethylamine. No need to know the structure, just remember the names over here. Okay, so mercaptoethylamine is the tail part over here. And if the question says that to which part of the coenzyme A does the groups are the groups attached, or which is the active part of the coenzyme A, right? So you have to say it's the mercaptoethylamine because it is the one which contains the thiol group, right? Or the sulfhydryl group. So this is the tail. Okay, tail is very important over here. Usually tails are not important, but here tail is very important. That's why I mentioned it first, right? So this is the body. The body is of, what is it? So body, it is the organic part of it because it's a coenzyme, right? So it is the organic part of it, which is nothing but the vitamin B5, pantothenic acid. Okay, so this is the this is the body part of it. And then it has the head part, which is a phosphorylated ADP, right? It is which is a nucleotide, simply a sugar, phosphate, and the base. Sugar, phosphate, and the base. That is all you need to remember about the coenzyme A. In the structure, you will remember it has a head, so this is its head, right? And remember the tail is important here for the coenzyme A. Okay. Do we have any questions about it? Did you all understand the function of the and the structure of the coenzyme A? All clear. Okay. Okay, so there's the two most important carriers of the hydrogen ions and electrons are NADH and FADH2, which is these two are upcoming in the TCA cycle. Okay, and we spoke about the vitamin part, right? Because they're coenzymes, so they have the organic part. So vitamin part in the NAD positive is the nicotinamide. The vitamin part of the FAD is the riboflavin. Okay, and the vitamin part of the coenzyme A is the vitamin B6, which is the pantothenic acid. Sorry, vitamin B5, which is the pantothenic acid. Okay, that was about it. And now let's watch. Wait a minute. Wait a minute. What happened? Can you all hear the noise? The sound of the video? Can you all hear the sound of the video? Okay, alhamdulillah, good. Wait a minute. What happened to it now? I'm not able to play it now. Okay, the voice is very low. Okay, give me a minute. Let me see how I can help you.
How do you turn that bite of food into a chemical that a cell can recognize and use as energy? The first step is altering the food into its component chemical compounds and then getting those molecules into your cells. That process is called digestion. Once inside your cells, the process of turning that bite of food into useful energy by cellular respiration begins. The process of digestion results with carbohydrates and other molecules being removed from the consumed food and transported into the bloodstream. From there, nutrients, like the carbohydrate glucose, will leave the bloodstream through a capillary wall and enter a tissue cell. Once inside the cell, cellular respiration will completely oxidize the glucose molecule, releasing high-energy electrons. The overall goal is to make ATP, a storage form of energy for most cells. Cellular respiration is a four-stage process that begins with glycolysis. Glycolysis literally means splitting sugars, and it is the first step of cellular respiration, occurring in the cytoplasm of the cell. Glycolysis consists of two distinct phases, an energy investment phase and an energy harvesting phase. In the energy investment phase, two ATP molecules transfer energy to the glucose molecule, forming a 6-carbon sugar diphosphate molecule. This molecule splits, and the energy harvesting phase begins. During this phase, the two 3-carbon molecules are converted to pyruvate, and ATP is formed. Glycolysis is a 10-step reaction that involves the activity of multiple enzymes and enzyme assistants. In the process, a net of 2 molecules of ATP, 2 molecules of pyruvate, and 2 high-energy electron-carrying molecules of NADH are produced. When oxygen is present, the pyruvate molecules and NADH enter the mitochondria, and the next stage of cellular respiration begins. The next stage of cellular respiration involves the movement of pyruvate into the mitochondria, where it undergoes oxidation. Each pyruvate molecule is converted into a compound called Acetyl-Coa. In the process of pyruvate oxidation, electrons are transferred to NAD, producing NADH, and a carbon is lost, forming carbon dioxide. The next stage is the citric acid cycle, also called the Krebs cycle. Here, Acetyl-Coa will bind with a starting compound called Oxaloacetate, and through a series of enzymatic redox reactions, all carbons, hydrogens, and oxygens in pyruvate ultimately end up as carbon dioxide and water. The pathway is called a cycle because oxaloacetate is the starting and ending compound of the pathway. For every glucose that enters glycolysis, the cycle completes twice, once for each molecule of pyruvate that entered the mitochondria. During pyruvate oxidation and the citric acid cycle, a net of 8 NADH, 2 FADH2, 2 ATP, and 6 CO2 are produced for each glucose molecule. In order to understand how the majority of the energy is produced by aerobic respiration, we need to follow the NADH and FADH2 molecules to the next stage, the electron transport chain. The electron transport chain is a series of membrane-bound carriers in the mitochondria that pass electrons from one to another. As the electrons are transferred between the membrane proteins, the cell is able to capture energy and use it to produce ATP molecules. Proteins in the chain pump hydrogen ions across a membrane. When the hydrogen ions flow back across the membrane through an ATP synthase complex, ATP is synthesized by the enzyme ATP synthase. Oxygen acts as the terminal electron acceptor. By accepting electrons, oxygen is reduced to form water, a byproduct of the electron transport chain. All the high-energy electron carriers from the previous stages of cellular respiration bring their electrons into the chain. From this, the bulk of ATP from the entirety of cellular respiration is produced, a net of 32 to 36 ATP. In summary, we have seen how the 4 stages of cellular respiration are responsible for converting the energy found in the glucose molecule into ATP, the energy battery of the cell. On average, 36 ATP molecules are produced per glucose molecule that entered the cell. In the process of producing ATP, oxygen is brought in from the bloodstream to be the final electron acceptor in the electron transport chain, and the carbon dioxide that is produced as a byproduct is released. The goal of cellular respiration is to transfer the energy from the food that we eat daily into ATP that our bodies can use. This process starts with the eating of a snack or meal and ends with capturing the energy from the complete breakdown of the nutrients into energy and carbon dioxide.
Yes. Did you all understand anything? Just a bit. I'm sorry about the volume. I was not able to increase the volume. I know it was too much, but I just wanted to give you an overview because what I remember when I was taught biochemistry, nobody explained to me why we are learning all these processes, why we are learning TCA cycle, what is the purpose of it. When my instructors taught me, because they suddenly used to start, okay, we are going to study today glycolysis, but why we are studying glycolysis? Right? Why we are studying TCA cycle? I was never taught it this way. So I myself has experienced what I faced when I used to study biochemistry. So that is why I first of all want you all to know that why are we studying all this? What is the basic aim? Right? What is the basic aim of okay, the sound suddenly got higher. Okay, good, alhamdulillah, Mustafa. Right? So what is the basic aim? Okay, I don't want you to haphazardly go and start, okay, this is TCS cycle, this happens in it. No. This is, but why are we studying TCS cycle? What is exactly is happening, right? So of course you have to know every bit of TCS cycle or electron transport chain, right? But glycolysis is not there because that was in the last chapter and we have omitted that chapter from the syllabus. Okay, so let's start first of all trying to. So this was about the video, and now I'll explain you step by step what we are going to study in the class. So please pay attention, very important, because the entire chapter is based on these 2 cycles. Okay, so basically you have to understand that when we eat the food, what happens to the food? Why do we eat food, right? So ultimate aim of eating food is that whatever energy bonds are present in the food, whether we eat fats, whether we eat carbohydrates, whether we eat proteins, right, all these are hydrolyzed, right? And they are converted into, for example, fats are converted into fatty acids, carbohydrates into monosaccharides, proteins into simple amino acids, right? So then finally, what is the ultimate aim? The ultimate aim is to produce the energy. That is what we're looking for. After finishing this chapter, you will see that at the end we will be able to produce ATP. That is the aim of this chapter. The catabolism, right? We are doing catabolism in this chapter, not the anabolism. We're doing catabolism. So we are going to break the food, break the energy present in the food, so that we will produce the ATP. Okay, that is the aim of this chapter. Point number one. Okay, now, okay, no, Mohammed, I don't want you to go into acetyl-CoA at the beginning. No, that is not the beginning. As I said, that we have to go step by step. So step one is that we are going to produce the energy in the form of ATP. Now, how do we do that? We usually do that in four different processes, as it was there in the video as well. But out of four processes in this chapter, we are going to talk about only the last two processes. Okay, this whole chapter is based upon two things: one, the TCA cycle, and the last one, the electron transport chain. Okay, but how do we reach to last two processes? Okay, let me give you a little bit introduction about the beginning. So once we have the glucose, okay, stage one, we're talking about the cellular respiration. Okay, so stage one is the glycolysis. As I said, that chapter is not here. That chapter was the last chapter which we omitted. Okay, so stage one is the glycolysis, which will take place in the cytoplasm of the cell. And what do we get in the glycolysis? At the end of the glycolysis, we get basically the pyruvate. Okay, that is the end. So the glucose which you had, okay, the food which you had, which will be converted into pyruvate. So that will be the stage one. Okay, so that is not we're going to talk about in this chapter. Now, what will happen to this pyruvate, right? This pyruvate, okay, will enter into the mitochondrial matrix, okay, and this pyruvate will be converted into acetyl-CoA. This is what we are talking about. So we're going to start from the acetyl-CoA. So we had glucose. Glucose was converted into the pyruvate or a pyruvic acid. Okay, so this was glycolysis, which we are not doing in this chapter. So this was stage one. In stage two, pyruvate was converted into acetyl CoA. Acetyl CoA. Okay, acetyl CoA. So this was your stage one, which was glycolysis. This was your stage two, which was conversion of pyruvate into acetyl CoA, right? And now, then what happens to this acetyl CoA, right? This acetyl CoA, right, will enter into the TCA cycle, which we're going to talk about in this chapter, right? Which is stage three over here. So stage three is you will convert these acetyl CoA, which will enter into the TCA cycle. And what is the end product here? The end product will be producing the NADH, FADH2, ATP, right? And once we have NADH, FADH2, then the last stage will be that these NAD or FADH2, which are produced here, they will enter into the electron transport chain, which we will discuss in today's chapter. And in the electron transport chain, that will be the fourth phase where we will produce the ATP. So this is our aim now. So where is where are ATPs produced? Some ATPs are produced in the Krebs cycle as well. Okay, but like most of the ATPs are produced in the electron transport chain at the end, which is in the stage four. Did you all understand the overview? Stage one, glucose to pyruvic acid. That is a glycolysis. We are not doing that in this chapter. Stage two, pyruvic acid to acetyl CoA. This stage two. We are not even doing that in this chapter. So we are going to discuss stage three and stage four in this chapter, right? So that is why we are going to start with the acetyl CoA. So that is why I told somebody said about acetyl CoA. That's why I told him don't say about acetyl CoA because they don't know where did this acetyl CoA come from, right? But now you know that we got this acetyl CoA from the pyruvic acid, from the pyruvate, and we got this pyruvate from the glucose. Are we all okay now? Okay. Now the locations are very important. You are definitely asked about the locations, right? So, for example, if we talk about the this the conversion of acetyl CoA into the NAD and FADH2. So this will take place in the mitochondrial matrix. Okay, so this process will take place in the mitochondrial matrix, while as the electron transport chain, which will take place in the mitochondrial inner membrane, right? So if you remember, I showed you last time we discussed about the mitochondria, right? So mitochondrial matrix is this one, right? So here we will have the TCA cycle will take place over here, right? And then we have the inner mitochondrial membrane. There we will have the the electron transport chain is present over in this inner mitochondrial membrane. Okay, so one by one we'll discuss about all this in detail. Let's start with the TCA cycle. Okay, so after understanding the overview, what is a TCA cycle? Okay, first of all, let's try to understand, because some students usually get confused. What is the TCA cycle? What is the citric acid cycle? What is the Krebs cycle? Remember that all of them are similar. You can either say call it a Krebs cycle, right? Because it was discovered by a person whose name was Hans Krebs, right? So it's called as a Krebs cycle, or it is also called as a citric acid cycle, right? Or it is also called as the tricarboxylic acid cycle. So don't get confused with the names. All of them means actually, all of them have the similar name. Now, what are we going to do in the acetyl? What are we going to do learn in the acetyl-CoA? I mean, what are we going to learn in the Krebs cycle? What is the goal of the Krebs cycle? Okay, what are we going to do with this acetyl-CoA? Okay, we have the acetyl-CoA. What are we going to do with this acetyl-CoA? Okay, this acetyl the result or the goal of the acetyl CoA of the Krebs cycle is that we will take this acetyl CoA and break down it throughout this cycle, throughout 8 steps. Okay, break down it, right? And then the energy which is stored in the bonds, that energy will be released, and that energy will be stored in these molecules, whether it is the NADH or FADH or the GTP. Okay, that is our ultimate aim here. We are looking forward to extract the energy, which will be in the form of these hydrogen ions, okay, and store that energy in the NAD positive or the FAT positive, and they eventually get converted into NADH or the FADH2. Okay, and you will see that this NADH or FADH2 will be used in the next step called as an electron transport chain to produce the ATPs. Are we okay? Did you all understand why we are studying the Krebs cycle? What is the goal of the Krebs cycle? What do we want at the end? Did you all understand? So what are we looking forward? We are looking forward for the, yes, NADH, FADH2, so that we can take these electron carriers to the next stage and extract the ATP from it. How we will use them for the ATP extraction, that I will teach you in the next step. Now, after knowing that what is the goal of the Krebs cycle, let's try to first of all see that what is the result of the Krebs cycle. What do we get? Before we discuss the Krebs cycle, this is the Krebs cycle, but let's try to discuss what do we get at the end. Let me erase all of this first. Okay, as I said that there are 8 steps in the Krebs cycle. Okay, and at the end of the Krebs cycle we will get NAD, we will get 3 molecules of NADH. Very, very important. All these things are asked in the exams. How many molecules, when they are produced, at which step they are produced. So very important, right? So we will get one is here, NADH, another NADH is here, and another NADH is here. So 1, 2. So how many NADH we'll get at the end? NADH. We will get 3 molecules of NADH. Correct? Okay. What about FADH2? How many molecules of FADH2 we have? Let me get the red color. We had only 1 molecule of FADH. So at the end of my Krebs cycle, I should have 1 molecule of FADH2. So 1 molecule of FADH2. What else do we get at the end of Krebs cycle? Look at this. I have at the end of it, I have GTP. What is GTP? GTP is like an ATP. The way we have adenosine triphosphate, here we have the guanosine triphosphate, right? So this will be later on converted to the ATP. So think that this is an ATP. This is the energy. Okay. So GTP. How many GTPs, ladies and boys? 1 GTP, isn't it? Yes or no? Okay. So this is what we are going to get in the form of energy at the end. Let me get a pen here. So this is what we are going to get at the end. Now what you need to understand here, did I explain that island yet? I am just trying to tell you that what do we get at the end. Okay, we are going to understand what do we get at the end. Okay, so just we will go step by step. The next step which is coming, that we'll learn how it is done, right? The next step is coming up, how. Okay, as I said that we first of all try to know what are we getting after, what are we going to get after the Krebs cycle, what are we looking forward for, why are we studying Krebs cycle. So how many NADH? 3 NADH, 1 FADH2 and 1 GTP. Okay, now what happens is that every time a molecule of acetyl-CoA enters, okay, so we have this acetyl-CoA. Basically, there were two acetyl-CoA which were produced. Okay, so one acetyl-CoA will enter, and all this will be produced. Then one more molecule of acetyl-CoA will enter, then all this will be produced. So can I multiply this with two? We will not talk about ATP now. We will talk about ATPs at the end, how to convert NADH into ATPs. So can I say that at the end of citric acid cycle, I will get 6 NADH, 2 FADH2 and 2 GTPs? Can I say that? Yes. Why do I say that? Because I don't have only one molecule of acetyl-CoA entering into the cycle. As I said that I have two molecules of acetyl-CoA entering into the citric acid cycle or the Krebs cycle. And why do I have two molecules of acetyl-CoA? Because initially when the glucose was split, okay, if you remember when I told you glucose was split to produce the pyruvic acid, right? So at that time when glucose was split, it produced two molecules of pyruvic acid, right? So two molecules of pyruvic acid were produced. So eventually what will happen? So you will obviously have two molecules of acetyl-CoA. That is why, remember, sometimes when the question is asked, read the question. Is it asking you per molecule of acetyl-CoA or is it asking you two molecules of acetyl-CoA? And if it's not mentioned there in the question, right, remember that you have to remember, you have to always multiply it with the two, right? Unless it's mentioned in the question. So remember, it's the two molecules which enters into the acetyl-CoA. So you will multiply it with the two. If the question says that write how many NADH are produced from the acetyl from the TCA cycle or the Krebs cycle, you should know that we have two molecules entering here. So you will not write 3 NADH, you will write 6 NADH, 2 FADH2, 2 GTPs. Did I make it clear? Everybody? Okay. Now, what else? As I said, that what is produced at the end of the cycle is very, very important. What else do we produce in the TCA cycle? Okay. Do you see here carbon dioxide? Look at this. We are also producing two molecules of carbon dioxide at the end. Okay. So we also produce H2O, not shown over here, but we also produce water here, right? So two molecules of CO2 are produced at the end. And do you know which CO2 is this? The CO2 produced in the Krebs cycle will straight away go to our lungs and we are going to throw that out from the lungs. So the carbon dioxide which you released from your mouth, it actually came from which cycle? Sometimes the question says like this. The carbon dioxide which we exhale out comes from dash. It comes from which cycle? Glycolysis? No, it comes from the, very good, Krebs cycle. Very good. Okay, remember that. So how do we get this carbon dioxide? You will see that the carbon dioxide which is, you know, which is released over here, it's the acetyl group. Okay, this one will be converted into the carbon dioxide. Okay, so are we okay about it? Any questions till now? No questions, all clear. Okay, now let's start this. Now this is the cycle. Now I cannot help in it, you don't all I can help you is that, okay, don't know the structures, don't memorize the structures, but yes, you have to memorize the enzymes, and I'll let you know how to I have a mnemonic for that as well. So let me finish with the Krebs cycle, then I'll explain you how you have to learn this. Yes, Noor, absolutely. So we have two molecules of CO2. Overall, if two acetyl-CoA enter, so if we will have how many CO2? We'll have four CO2. Yes, Noor, very good. Absolutely. So you have to, sometimes it varies. Sometimes the question says just one molecule of acetyl-CoA. So sometimes the question says a general one. How many molecules of CO2 are produced in a citric acid cycle? So at that time, if it's not given in the question, you have to assume that it has to be twice. Sometimes the examiner wants to check you that whether she knows that two molecules of acetyl-CoA enter into Tca cycle. So it depends upon what kind of question is asked there. Okay. If it's not mentioned over there, then assume that you know that it's the two molecules which enter into the Tca cycle. So that way you'll act smarter. Okay. Okay, now how many steps we have? We have 8 steps over here. Okay. And what do we start? Okay, you have to know the starting point as well. Okay, what do we start with? We start with a molecule called as an oxaloacetate. This is the one called as oxaloacetate. Now, what you need to know is that people might ask you about, or examiner might ask you about the oxaloacetate. We are more interested whether you know how many carbons are there, because knowing about carbons is very very important in the TCA cycle, right? So you should be knowing that oxaloacetate, when we start with the oxaloacetate, okay, oxaloacetate is a 4 carbon compound. Can we see it here? 1 2 3 4, right? So 4 carbon compound of oxaloacetate combines with the acetyl-CoA. Okay. So the starting point in the cycle is this acetyl-CoA, which is a 2 carbon compound. Look at this. I have 1 and 2 carbon only here. So these two are condensed together to form 4 plus 2 is how much? So I'm going to write the carbons here so that you don't forget it, to remember about the carbons. So 4 carbon, 2 carbon here. What do we get at the end? What is produced, guys? Citrate. Very good. How many carbons? 1 2 3 4 5 6. I'm more interested that you should be able to know that 4 plus 2 carbons were condensed together and we produced a. Sometimes the question says that how many carbons are present in the citrate? Okay, so you have to know that there are 6 carbons present in the citrate. And enzymes, of course, we have to remember the enzymes. The enzyme is the citrate synthetase enzyme because it's synthesizing the citrate. So it's the citrate synthesizing enzyme over here. Okay. Now what happens? This citrate, now please pay attention everyone, this citrate is converted into the isocitrate. So nothing is happening over, it's just the isomerization reaction. There'll be no change in the carbon. I will still have 6 carbon. This was simply an isomerization reaction over here, right? So citrate was converted into isocitrate, and the enzyme here is the aconitase. So we'll first learn, know the cycle, and then we'll go step by step once again, okay? So once the isocitrate is produced, which is again a 6 carbon compound, okay, this isocitrate will be further converted into the alpha ketoglutarate. This will be oxidized to the alpha ketoglutarate. Okay. And what is the enzyme here? We have the isocitrate dehydrogenase. Okay. And remember that this is the step, very important. This is the step where the first molecule of carbon dioxide is produced. Okay, sometimes the question says that Name the step when the first molecule of carbon dioxide is produced. So the question says that, if the answer says that it's from, it's the step 3 when the isocitrate is converted into alpha ketoglutarate. This is the place when first molecule of CO2 is produced. And this is also the step where the first molecule of NADH is produced over here. Are we okay till now? Do we have a carbon change here? Do you think how many carbons are there in the alpha ketoglutarate? We removed one carbon dioxide. So obviously we lost one carbon over here. So alpha ketoglutarate is just a very good 5 carbon compound. Okay. Now further decarboxylation will take place. Okay, decarboxylation will take place, and the second molecule of O2 is going to come out at the step number 4, wherein the alpha ketoglutarate will be converted into the succinyl CoA, right? And one more molecule of carbon dioxide came out. So what can you expect about a succinyl CoA? Succinyl CoA is the 4 carbon compound, because one more CO2 came out. So these two steps are essential, because this is a step number 3 and step number 4, I think, right? Why they're essential? Because these are the two places where carbon dioxide is coming out, and these are the two places where your NADH is also formed, which we are looking for. We're looking for NADH, right? So that's why I said that these two steps are very important. Step number 3 and step number 4. Okay. Sometimes they just ask you the name the step. Carbon dioxide is released, then the options might be given from step number 1, step number 8, step number 5, step number 3, right? Sometimes just the steps are given. Not necessarily they will write isocitrate to alpha ketoglutarate or alpha ketoglutarate to a succinyl CoA. No. Sometimes they just write about the step. So remember the steps as well. So step 3 and step 4 is the place where carbon dioxide is released. Step 3 and step 4 is the place where NADH is formed. Isn't it? Till now. Step 3 and step 4 as well. So till now, we are still going further. Okay. Are we okay till now, everyone? I'm just going as slow as possible. I'm just reducing my pace. Yeah. I will repeat. Okay. Yes, Gadir, don't worry. I just said that. Yes, no problem, no problem. I just said that. Excuse me. I just said that the when we sometimes they don't ask you, uh, you know, at which, sometimes they simply ask you at which step of the Krebs cycle carbon dioxide is released. So you have to remember by default, it is the step number 3 and the step number 4 where the carbon dioxide is released, right? And it's the step number 3 and the step number 4 where your NADH is also formed. Look at this. Look at this. My NADH is formed in the step number 3 and step number 4. Right? So that's what I said that. So just quick recapitulation what we said. We said that we had four carbon compound called as an oxaloacetate, which condensed with the 2 carbon compound called as an acetyl-CoA in presence of the enzyme citrate synthetase, and we got a 6 carbon compound called as the citrate. Okay. Then the citrate was converted into isocitrate. It was just an isomerization reaction in presence of the enzyme called as an aconitase. We still have the 6 carbon compound called as an isocitrate. And then the changes will start happen, right? What are the changes? From isocitrate, 1 carbon compound, 1 carbon dioxide will be released, right? And it will form a 5 carbon compound called as the alpha ketoglutarate. Okay. And while isocitrate is converted into alpha ketoglutarate, we also have one NADH produced over here. Okay. So once we discuss these steps once again, in the forthcoming slides, so you'll get to know how this NADH was produced over here. Okay. Then finally, we have a decarboxylation reaction once again here, where one more carbon dioxide was released here. So this is the step 3 and step 4, so you don't get confused. So this is step 3 and step 4, right? So one more carbon dioxide was released over here. And as the carbon dioxide was released here, what happens? We also got one more NADH here. And now we have a 4-carbon compound, which is a succinyl-CoA. Now, after this, there's no change. Remember that after that, there is no change in the number of carbons. So remember that. So no change after this. So that's how you can, memorize it. No change in the number of carbon atoms after this, after succinyl-CoA. Okay. Succinyl-CoA will be converted into the succinate. As you can see here, this is a succinate over here, right? So this is the place when your GTP is produced, or you can say the energy is produced. So if the question says that GTP in the Krebs cycle is generated in which step? So you have to know that it's generated when you convert this. Just remember the names. No reactions when you convert the succinyl-CoA into the succinate, right? And you can also remember that it's the step number 5. Absolutely. Remember the steps as well. Now, after that, I have succinate. Okay. This succinate will be converted into the fumarate, and this is the place when the FADH2 is released. So we can remember that FADH2 is released when the succinate is converted into fumarate. So no change in the carbon, because after this you will see that no carbon dioxide is released. So this is also 4 carbon, this is also 4 carbon, right? So malate is also 4 carbon, right? Till we reach the oxaloacetate once again. They are all 4 carbon compounds. Yeah, we'll discuss Muhammad one by one as we discuss that. We'll discuss about that, the oxidations and the decarboxylations. Okay. So once we have, so this was the place where your FADH2 was released. And after that, you have to remember the names. Okay. Fumarate is converted to the malate. And here, if you see here, the water is also produced here. This is the place where water is produced, right? So fumarate is converted into the malate, and the enzyme here is the fumarase. Okay. Now, after that, the malate is converted into the oxaloacetate in presence of the enzyme malate dehydrogenase. Okay. And this is the place, right? This is, I think, step 8 or step 7, it should be, right? What is it? Is it step 7 or step 8? If this is your 3, 4, 5, 6, 7, it should be step 8, right? Yes, I think step 8. Yeah. So at step 8, I have my one more NADH produced, right? So you have to remember that NADH is produced at step number 3, step number 4, and step number 8, right? Okay. And carbon dioxide is produced at step number 3 and step number 4. And GTP is produced when? This is 4 and this should be 5. Yeah, this should be 5. So GTP is produced at step number 5. Let me write it here. So step 5, GTP is produced, right? And what about the FADH2? Who can tell me about the FADH2? Right after this, we have FADH2. So obviously FADH2 is the step 6. So remember these steps. You can either remember them as steps. Sometimes, as I said, that questions might be asked based on either the steps or based on the what is converted into what, like, right? Did you all understand till now? We are going to go over with it once again, step by step in the upcoming slides now. Okay, yes, Yomna. I said that NADH is produced at step number 3. Let me put a at step number 3, if you see here. Okay, at step number 4, NADH, Yomna, and step number 8, NADH, right? And carbon dioxide, step number 3 and step number 4, only carbon dioxide. GTP, step number 5, okay? and FADH2, step number 6. Okay, Yamna? Okay, great. So do we have any questions? Now, there's no shortcut here. You need to know the enzymes. You need to know, okay, about the 8 key molecules produced here. But don't worry, I'll give you a mnemonic. The mnemonic is coming in a while. Okay. So let's discuss steps one by one. Okay. So step one, if you remember step one, what did we say in step one? We said that the 4-carbon compound oxaloacetate combined with the acetyl-CoA, which is a 2-carbon compound, and we got the citrate at the end. We got citrate at the end. So what is happening here in this step? Okay. As you can see here in this step, we have the enzyme citrate synthetase here. Okay. And if you see here, what is getting, what is happening here? Anybody who can explain this step? What is happening in this step? Remember acetyl-CoA, we spoke about that. Anybody who can explain what is happening in this step? Nobody? What is it? If you see here, this acetyl-CoA is attached over here. Can you see it here? This is my acetyl-CoA, right? It forms the citryl-CoA. This is an intermediate compound with the 2 carbons. Yes. So if you see here, yes, there's a group transfer over here, right? So it got attached over here, and then one water came out. Excuse me. Sorry, water was added, and then what did we get? We get the condensation reaction. We got the citrate over here, right? And what you need to remember here is that this step, what causes the inhibition of this step? Okay, so this step can be inhibited, or this step cannot go forward if we already have in the cell too much of NADH, too much of ATP, or too much of succinyl CoA, right? If these things will be there in the step in the cell, of course, then no need to if there are enough ATPs in the cell, enough NADH in the cell, so obviously this is a rate limiting step, then it will not go further. Okay, it will not go further. Okay, so it's going to stop over here, right? So presence of NADH, presence of ATP, or presence of a large amount of succinyl CoA can inhibit this step. That means it will not go further. Okay, any questions? So here what did we have here? We had oxaloacetate, which combined with the acetyl-CoA, right? And then what did we get? We get this, look at this. This was attached here. So we got the citryl-CoA. And then what happened after that? After that, there was a cleavage, right? So, and then we got the citrate over here. Okay. So this was simply a hydrolysis reaction. In step 2, what happens in step 2? Step 2, if you remember here, what was, let me erase all this first. What is happening in this step 2? Everyone, we have a citrate, and citrate is converted into what? It's converted into the isocitrate, right? So if you see here, this is my citrate, okay? So what happens? Water comes out here. Look at this. Water comes out here. See, this OH has been removed. That is why we got a double bond over here, and one H was removed from here. Okay, so we removed this OH and this H. So dehydration reaction. Okay, so we got the aconitate. Okay, then rehydration. One more hydrogen was added here in presence of the enzyme aconitase, and we got here isocitrate. We'll not go further into detail. That is all we have here, right? From citrate to isocitrate, there's no carbon change here. Okay, only if you see here, the molecules were rearranged because the dehydration took place first and the rehydration took place here. Are we okay till now? Now, out of these three molecules, if I see here, this is also symmetrical, isn't it? This and this is similar. So of course, this is not chiral, right? So this, there's no question of having chirality here because I have a double bond over here. So this is achiral, this is achiral. Out of these three molecules, only isocitrate is chiral with the two what? With the two chiral centers, right? So of course we'll have four stereoisomers present over here, but out of four stereoisomers, only isocitrate is possible. Because if you remember, I told you even if we have plenty of stereoisomers in the body, only one will be active, one will be used in a particular reaction. Otherwise, there will be a lot of confusion in the body, right? Did we understand the step number 2? What is happening here? Simply dehydration and then rehydration. Molecules are rearranged. Okay. Step number 3. If you remember in the step number 3, what did we say in the step number 3? In the step number 3, we said that what happened? We had isocitrate, right? Isocitrate was converted into alpha ketoglutarate. And I told you this is an important step. Why? Because this is the step where your carbon dioxide is released and you also form the NADH, right? So isocitrate, as you can see here, so there will be an oxidation which take place. Isocitrate will be oxidized, okay, as you can see here, using the NADH, okay, using NADH, isocitrate is oxidized, and then what do we get? And then if we see here, then we get oxalosuccinate over here. You don't have to know the name of the intermediate compounds. Then this carbon dioxide will be removed from here. So decarboxylation reaction. And then at the end, what do we get? We get the alpha ketoglutarate. So how many carbons we have in the alpha ketoglutarate? We started with what? We had 6 carbons, and then we have only, as I said, that this point is very important. How many carbons we have? Only 5 carbons here. Okay, so we don't have, as I said, that what is important here is you have to know, in this process we produced NADH, and you will see that this NADH will be used later on. Okay, so this was the step 3 where we formed the alpha ketoglutarate with the decarboxylation reaction. Then comes your step 4. In the step 4, you have the alpha. If you remember, if you go back to the cycle, what happened in step 4? The alpha ketoglutarate was converted into the succinyl-CoA, right? And one carbon dioxide was removed, one more carbon dioxide was removed, and we also produced the NADH. Let's go to the step 4 here, right? So we had alpha ketoglutarate, which was a 5 carbon compound, right? And then what happened over here? Anybody? So we have a decarboxylation reaction, oxidative decarboxylation reaction. Look at this. We have NAD converted into NADH, and at the same time, we have this, what? This CO2 is released. Okay, look at this. This CO2 is released. So oxidative decarboxylation happened over here. Okay, and once we have the succinyl-CoA, how many carbons we have in the succinyl-CoA? Because another molecule of carbon dioxide was released because of the oxidative decarboxylation. Right, so this is the step 4. Okay, so moving on further, what happened then? After that, the steps are very simple. Okay, in step 5, what is going to happen? We had the succinyl-CoA, right? What will happen to succinyl-CoA? In presence of the succinyl-CoA synthetase, we have the succinate which is produced over here. Okay, and at the same time, we have the GTP produced over here, right? So what happens in the step 5? In the step 5, the GTP is produced here, right? And if you see here, the acetyl CoA was cleaved from here. It was cleaved from here, right? So this is called as an energy producing step. If the question says that which is the energy producing step of the Krebs cycle, so you have to know the energy producing step is the step 5, where your succinyl CoA is converted into the succinate. This is an important point. And you have to remember that this is the first energy producing step, because before this we did not produce any ATP, though we produced NADH, but NADH is later on used to extract the ATP, right? But here we directly produced the ATP in the form of the GTP. So that is why we say that this is the first energy producing step. Okay, is it clear? So what did we get at the end? We got a GTP and we got the coenzyme A, and then we got the succinate, right? This you have to remember. GTP is the energy, as I told you. GTP and ATP is, right. GTP is guanosine triphosphate. ATP is adenosine triphosphate. The base is different over here. Okay. Now what happens? Okay. Now we have the step 6 here. Okay. In the step 6, what's happening? Okay, we have the dehydrogenation will happen here. That is why we have the FADH2 produced from the FAD. Okay, as you see, the because of the dehydrogenation reaction, or you can say the oxidation of succinate. From succinate we will get the fumarate over here. Okay, so in short, your succinate was dehydrogenated to the what? To the fumarate by the FAD. Okay, by the FAD. So we have FADH2 produced over here. Okay, guys, are we okay till now? Everybody is so silent. Are we okay? Are you able to understand? See, you don't have to pay attention on the reactions. As I said that what is coming up, knowing the enzymes, knowing the products, knowing the key products, knowing when is carbon dioxide released. So all these things are very important. You don't have to pay attention on the reactions will not be there. Okay, so step 7 is the hydration. What happens in the hydration? In the hydration we simply add a molecule of H2O. Okay, so when we add a molecule of H2O, okay, so we add an OH group here. Okay, so as we add an OH group over here in presence of fumarase, we form the malate. Okay, and if you see here, by adding the OH group, the double bond was broken, and now we have a single bond here in the malate. So breaking of the bond will take place by the addition of the OH in the form of H2O to the fumarate. Okay, that was a step 7. And then the last step was a very important step, is that production of the oxaloacetate. Sometimes the question says that when do we regenerate the oxaloacetate? Okay, so when do we use the when do we produce the oxaloacetate? Sometimes this question is very, very important. Okay, so what is important, that at the end oxaloacetate, if you remember, we started with the oxaloacetate. If you remember here, we started with it was a starting molecule over here. It was a starting molecule over here, oxaloacetate, right? So what happened in the oxaloacetate, that we were able to regenerate the oxaloacetate. Very, very important, right? So oxaloacetate will be produced again, will be regenerated once again at the end. Okay, and at this step we have one more NAD, which NAD positive, which will be reduced to the NADH. Okay, so if you see here, at the end we end up having a ketone. This is a ketone again, oxaloacetate, right? So we'll end up having a ketone. So oxaloacetate was a starting material. If you remember, we said oxaloacetate was combined with the acetyl-CoA, right? So oxaloacetate is regenerated at the end of the cycle, okay, which will again combine with the acetyl-CoA, and the cycle will continue and continue and continue and continue and continue. Are we okay till now? Mohammed, what is produced over here? What is produced? No, no, not in, it's produced over here. Okay. Sorry, water is added over here, not produced, it's added over here. Here. So we did the hydration reaction. No more water is added over because we did the hydration reaction over here, right? We added the water molecule to the fumarate. So water is added over here, not the produced. Water is added over here so that we can produce the malate. If you see here, we add the OH group over here to produce the malate. So water is added over here, Mohammed, not produced. Okay. So that is all you need to remember about the 8 steps of the citric acid cycle. Okay. So as we already discussed about it, we produced 3 NADH, we produced FADH21, right? And we produced 2 molecules of CO2. This is like per molecule of acetyl-CoA, remember that, right? And we produce 1 molecule of GTP, right? We are interested in these things. This is of our use, this is important for us, this is important for us, this is important for us, right? So we will take this NADH and FADH2 to the next level now, and then we will produce the ATP out of it, right? That we will do in the next part of it. Okay, this NADH and FADH2 we will use in the electron transport chain, which is the second part of this chapter. Okay. Do we have any questions till now? So can anyone tell me now? Okay, TCA cycle oxidizes acetyl-CoA to TCA cycle oxidizes acetyl-CoA to what is acetyl-CoA oxidized to? Can you name this? Yes. No. We started with the acetyl-CoA, right? So TCA cycle will oxidize acetyl-CoA to what? To the carbon dioxide, right? So it's the carbon dioxide. Yes, it's the carbon dioxide. It will oxidize acetyl-CoA to the carbon dioxide which we exhale out. Okay. So now another question. All enzymes of the TCA cycle are present in dash. Where are the enzymes of the TCA cycle present? Then see, you saw so many enzymes are there. Look at this: citrate synthetase, aconitase, right? So where are these enzymes present? Where are the enzymes of the TCA cycle present? It was not in the inner membrane. Inner membrane, we have the electron transport chain.
The mitochondrial matrix.
Matrix, absolutely. All enzymes of TCA cycle are present in the mitochondria, except one enzyme. Except one enzyme, which is this one, Cyanide Dehydrogenase, okay, which is present in the inner membrane, right? The rest, all of them are present, all 7 of them are present in the mitochondrial matrix. What is the outcome of the TCA cycle? What do we produce at the end of the TCA cycle? Ladies, boys, everyone. 6 NADH, 4 CO2. Okay, very good. 2 FADH2, very good. What about GTP? 2 GTPs, absolutely. Other than that, how many molecules of CO2? Yes, 4 molecules. Remember, per 2 molecules of acetyl-CoA. I'm just repeating this once again, okay, per 2 molecules. Okay. So now the question is how you will remember. That was a big question, basically, how you will remember. So what I did is like, so this is the same thing, one turn of the citric acid cycle, and then you multiply it with the 2, okay? So then this is the same thing, just leave this. Okay, this is, the mnemonic which can help you to remember the key molecules of the citric acid cycle. If someone has any other mnemonic, I would appreciate if you can share it with me so that I can share it with I can incorporate that in the presentation and upload the presentation on the Moodle, or share it with everybody on the Moodle, right? If you have anybody, if anybody will come up with any other mnemonic, okay? So, the important thing about the the 8 key molecules are: can I keep selling substances for money, officer? Right? So the first letter is like citrate, isocitrate, alpha ketoglutarate, succinyl-CoA, succinate, fumarate, malate, and oxaloacetate. This is a mnemonic to remember the 8 key molecules. Is it clear? But you have to remember that you start with the acetyl-CoA. So acetyl-CoA, you have to understand that we start with the acetyl-CoA. Okay. Now, how to remember the enzymes? Okay, so for the enzymes, the mnemonic is so at disco devil sip down 5 drinks. So you have to remember that it's, if you know the 8 key molecules, if you know that citrate, okay, is formed when the oxaloacetate and the acetyl-CoA combines, it forms the citrate. If you know about this, so then you should be able to simply, because the mnemonic does not talk about the first letter of the first word of the enzyme, right? It basically talks about the enzyme itself. So synthetase, right? So this is synthetase. At is a conitase. Okay, D is dehydrogenase. Okay, dehydrogenase, because this is the place when the NADH is produced. Okay, and then we have the synthetase, then we have the dehydrogenase, then we have the fumarase, and then we have the dehydrogenase again, right? So before learning about the enzymes, you have to know about these 8 key molecules. So you can simply add citrate and isocitrate and alpha ketoglutarate to it as per the 8 key molecules present in the citric acid cycle. Do you think it's going to help you all? Yes, ladies and boys. Okay. So yes, if you have any other mnemonic, as I said, that you can share it with me. Okay. So to sum up everything, I made this slide to sum up everything. What is the outcome? So during the reaction of the citric acid cycle or the Krebs cycle, okay, 3 important events took place. One, we produced the ATP in the form of the GTP. Okay, we produced NADH and the FADH2. Okay, and then we produced the carbon dioxide, right? And other than that, now we'll move on to the next one. But before we move on to the okay, this one first. Why is the production of NADH and FADH2 important in the Krebs cycle? Because the next part of this chapter, which we are going to discuss in a while, the electron transport chain, right? You will see that there we are going to use the same NADH and the same FADH2 which were produced here. They will be used to yield our ATP, to produce our ATP in the electron transport chain via the oxidative phosphorylation, which is coming up. Okay. So why is Krebs cycle called as a citric acid cycle? It's named after the 6-carbon citric acid molecule produced here, the citrate. Citrate can also be called as the citric acid. Okay, if you remember, we said that 4-carbon oxaloacetate combines with the 2-carbon acetyl-CoA, it gives a citrate. Citrate can also be written as citric acid, right? So it is a 6-carbon compound. That is why it is sometimes called as the citric acid cycle. Okay, is it clear till here? And then, okay, now we started with the acetyl CoA, right? We started with the acetyl CoA. Now, this acetyl CoA, as I told you that this acetyl CoA, we got it from the glycolysis, right? If you remember when we started, I told you glycolysis, we use glucose, which converts it into pyruvate, and pyruvate is converted into acetyl CoA, right? But if carbohydrates are not available, right? If carbohydrates are not available, we can still use the proteins and the fatty acids, right? They can also produce proteins and the fatty acids. They can also produce the acetyl CoA. If you remember, we spoke about using non-carbohydrate sources, non-carbohydrates as a source of energy. So they can also lead in the, when we do the beta oxidation of fatty acids, they can also form the acetyl CoA, right? And even proteins can be converted into the acetyl CoA. So it's not necessary that only your glucose will be converted to pyruvate and then to the acetyl CoA. So they can also join in between, right? And they can also form the acetyl CoA. Is that correct? Did you all understand? So it's not only the glucose which can form the acetyl CoA. Other non-carbohydrate sources can also form the acetyl CoA. Okay. And then these are the intermediates of the citric acid cycle which we spoke about. So NAD positive and FAD positive are the coenzyme that picks up the electrons in the form of hydrogen atoms, which we saw, right? And now you will see that these hydrogen atoms, right? The NADH and FADH2 will be, will be used, in the electron transport chain. Okay. So who is going to answer this easy question? When NAD positive and FAD picks up the two hydrogen atoms, what do they form? What does your NAD positive form and what does your FAD positive, what does your FAD forms? NAD forms NADH, right? And FAD forms FADH2, right? So this is like, now the next part of the chapter is the oxidative phosphorylation. But before we start, we'll just take a 5 minutes break because I don't want you all to get confused about it. Take a 5 minutes coffee or a tea break, and then we can start all over again, because that is again a next whole process where there are a lot of things happening there again. Okay. So next 1.5 hour, I think 1 hour, it might take us 1 hour to finish the oxidative phosphorylation. Okay. So are we okay till now? Do we have any question till now? So we'll take a break from 10:45 to 11:50. Okay. So you all can take a coffee, tea, whatever break. Okay. Then we can start with the next part of it. So meanwhile, if you have any question, you can discuss it with me, or if you have any concern till now. Yeah. Okay. So now, see, we are doing catabolic pathways, right? And I already explained you that we in this chapter we are doing step 3 and step 4, right? We are not doing glycolysis. We didn't talk about how acetyl CoA is produced, but we only spoke about because if you remember, I told you the aerobic respiration is divided into 4 phases. Okay, one was the glycolysis, second was the formation of the acetyl-CoA, third was the citric acid cycle, and this is the fourth phase of the aerobic respiration. And this is a very important phase because in this phase, we are going to produce the ATP. That is our aim, right? We are going to produce ATP in this part, right? So what is the purpose of we already spoke about what was the purpose of a catabolic pathway. We simply converted the chemical energy which was stored in our food, right? And we have to convert that chemical energy into ATP. So ATP production will take place here, right? So ATP production took place in the previous step as well, right? In the citric acid cycle as well, in the glycolysis as well. But like the maximum amount of ATPs are produced in the electron transport chain or the oxidative phosphorylation. Okay, so this is the second part of this pathway. Okay, or you can say not the second as per this chapter, but fourth as per the aerobic respiration. Okay. So what happens in this part, this is called as an oxidative phosphorylation. Okay. In this oxidative phosphorylation, it is divided into two parts. One, we will talk about the what is an electron transport chain. Okay. And then the second part, we will talk about what is chemiosmosis. So again, in this part, we have two, sub parts. One is the electron transport chain, and the second one is the chemiosmosis or the phosphorylation. Okay. Now, first of all, the question, where does this process take place? Okay. So we have the enzymes of our oxidative phosphorylation. They are not present in the matrix. They are present in the inner membrane of the, let me get my pen. What's wrong with it? They are present in the inner membrane. This is the mitochondria. They are present in the inner membrane of the what? Mitochondria. So here we're not going to talk about the matrix and all. Okay. Now, what is going to happen here? What is the main idea here? Okay. The main idea is in the TCA cycle, the NADH which we produce, the NADH and the NADH and the FADH2 which we produced in the citric acid cycle, okay, what will happen? They will enter into the electron transport chain, okay, and when they will enter into the electron transport chain, they will be converted into the ATP, okay. At the same time, you will see that they will also oxidize a molecule of molecular O2. We'll see that, right? So O2 will be the final electron acceptor, okay. So one by one, we'll go to that in detail, but I'm just telling you the main idea first. Now, you might be wondering, she said that the NADH and the FADH2 will enter into the electron transport chain. You might be having a question, what is an electron transport chain, right? So you have to remember that electron transport chain is nothing but we have the various or a series of electron carriers present in our inner mitochondrial membrane, in the cristae of the mitochondria. Because if you remember, the mitochondrial inner membrane is thrown into folds which we call as the cristae. Look at this. So in this membrane you will find the electron transport chain present. Now let me show you the diagram first. These will be the electron transport chains which we'll be talking about, right? So in these electron transport chains, you will see that they're all electron carriers. We call them electron carriers, okay? Because they can pick up the electron and give from one carrier, it is sent to the next carrier, then to the next carrier, right? In the chain. So this is if you see, this is the plasma membrane, right? And this is your inner membrane. This is the inner membrane of because they are all, the electron carriers are present in the inner membrane of the mitochondria. Okay, so I'm just telling you the main idea. The main idea is what will happen that as these electron carriers, they pick up the electron, they give up to the next one, then to the next one, right? So as the electrons move through these electron transport carriers, they release some energy. Please pay attention, everyone. So you have to understand the main idea. Okay, then we will go, step by step into it. So as the electrons, they move from one carrier to the other carrier, what are they going to do? They release some of the energy from from those electrons, right? And that energy is used to pump the protons, okay, into the inner membrane space. So that means you will see that, let me erase this here. You will see that at the end of electron transport chain, you will see there will be plenty of protons present in the intermembrane space. That is our, as I told you, before we start something, we should know why we are studying this. What are we aiming for? We are aiming for, number one, the electrons will be transmitted from one carrier to the next carrier to the next carrier. Point number one. And while doing so, we are actually pumping the protons from the matrix. The protons will be pumped through these protein carriers into the intermembrane space. As a result, at the end, we will have high concentration of protons over here. So you can say that the proton gradient will be established in the intermembrane space. As the proton gradients will be formed over here, then you will see that the final acceptor of the electron will be the oxygen, which will produce the water, right? And at the same time, these protons will go again, down their concentration gradient, and then at the end we will have the ATP produced. So again, your oxidative phosphorylation has been divided into two steps. First, we will talk about the electron transport chain up to here. Okay. So once we will have electron accepted by the oxygen over here, then the second step we will talk about how these protons will help us to produce the ATP. So again, we have two more steps which we have to discuss in this lecture now. Now tell me how many of you did not understand what are we going to do, what we are going to what is the uh, key step, or what are we going to learn in this lecture. That is as I said, that I want all of you to know why we are studying the oxidative phosphorylation. Then we will discuss what is oxidative phosphorylation. Am I clear? What is our aim? Are we okay? Did you all understand? What are we looking forward for? Any questions? Everybody is okay, right? So two processes which we are going to study now, which we are going to discuss now. As I said, that the first will be the electron transport chain, and second will be the phosphorylation, or this one we'll discuss little later. This is called as a phosphorylation or the chemiosmosis. We'll first discuss about the electron transport chain. Okay, we are all okay. Okay, are we okay now? Okay, so now what? First of all, we'll start with the where do we find these electron transport chain, right? We said that the electron transport chain is so this diagram is everything. I'll not go into step by step over here. I will explain you everything on this diagram because it's all about this diagram. Even if we have steps over here, right? Even if we have steps over here, if you concentrate on the diagram, you will understand everything from here itself. Okay. So we start with, as I said, that where is this electron transport chain present? That is my first question. Electron transport chain, which we also call as the respiratory enzymes. Okay, we have four types of the respiratory enzymes, which you can see here. This is your complex 1, right? This is your complex 2, this is your complex 3, and this is your complex 4. right? So these are your four respiratory enzymes, which we also call as complex 1, complex 2, and complex 3 and complex 4. Where are they present? They are present inside the what? In the inner membrane of the mitochondria. Now, what else do we have? Other than that, if you see here, we also have the COQ and then we have the cytochrome C. What are these? Okay. COQ and cytochrome C are nothing but they are simply the electron carriers. Okay, they are simply the electron. They are, for example, COQ is basically the hydrophobic carrier. When I say hydrophobic carrier, that means if you know the structure of the plasma membrane, right? We have phospholipid bilayer over here. When I say hydrophobic, don't you think so it can move here and there and here and there everywhere because it's hydrophobic in nature? COQ, can I say that? Yes or no? Yes, because that's why I said that the COQ is, which is also called as an ubiquinone. It is a hydrophobic. So in this carrier, you will see that it can move here and there anywhere. While as these complexes, no, they cannot move here and there, right? Because they are present within, they are like an integral membrane proteins. If you remember, we spoke about integral membrane proteins last semester, chapter number 3, right? So that is why they are stable at one place. This complex 1, 2, 3, and 4, they cannot move. The only person who is going to move over here will be either COQ here, and then one more electron carrier is the cytochrome C over here. So they are called as the mobile carriers, which are present in between these complexes. If you see here, COQ is present in between the complex 1 and complex 3, right? And cytochrome C is present between the complex 3 and the complex 4. Now, what is their function? Simply, you will see that their function is to transfer the electrons between the complexes because they can move here and there. Nobody is going to stop them because they all are hydrophobic in nature. Both of them are hydrophobic in nature. Are we okay? Can I say complex 2 cannot pump H2 into island? I didn't even explain that yet. I'm going to explain that once. I didn't even start explaining complex 1, complex 2, complex 3. We're just doing the introduction of complexes. I will explain that in a while once I reach there. Okay, island, right? We are going step by step. Okay, okay. Now let's start discussing about the complexes. Okay, so now we know that what is the function of these complexes, right? As I told you, they are the respiratory enzymes, right? And then I told you COQ and cytochrome C, they are simply the mobile carriers of the electron transport chain. Okay, and you will also see that this COQ will be used as an oxidizing agent. Okay, it can be, see, as I said, that COQ is, is a mobile carrier. It can move here and there. It can sometimes be associated with this, okay, the COQ, or this COQ can also be associated with this, okay, with the complex 2, right? So they can be used as an oxidizing agents, okay? Now let's start discussing about all these, okay? Uh, Before that, before we can explain the process about it, let's discuss about these complexes in detail, and then we will discuss how things will happen here. Then we'll discuss, let's discuss the anatomy first. We all, whenever we do anatomy and physiology, we all start with the anatomy and then discuss the physiology. Let's discuss the anatomy of these respiratory enzymes, complex 1, 2, 3, and 4. Okay. So as I said that all of them are collectively called as the electron transport chain. Okay. And I told you that they are also called as the respiratory enzymes. Okay. Now, if you ask me, out of these four respiratory enzymes, which enzyme is larger? Okay. or which respiratory enzyme is larger as compared, in these four, in the group of these four enzymes. So I would say that complex 1 is larger because it is made up of 40 subunits. As you see over here, it is made up of 40 subunits. Okay, this larger enzyme, what is present in it? Inside it, it has the flavoproteins. As you can see here, FMN is over here. This is the flavin mononucleotide. This is a mononucleotide, right? And it also has, okay, iron and sulfur attached to it. Iron sulfur protein is attached to it, right? So this iron and sulfur is attached to it. Okay, so FMN is an organic part. You know what is a coenzyme, an organic part, right? So this is used as a cofactor over here. Okay, so this is made up of 40 subunit, complex 1. Okay, then we talk about the complex 2. Okay, about the complex 2, what you need to remember here, this is the only complex, okay, which cannot pump the protons into the intermembrane space. If you remember, when I started explaining you about what we are going to do in this, I told you our aim is to pump the protons in the intermembrane space, right? So all these complexes, whether it's a complex 1, whether it's a complex 3, whether it's a complex 4, all of them have the channels present in between through which protons can go into the intermembrane space. The only respiratory enzyme which does not have the proton pumping towards the intermembrane space, that is your complex 2. Did you all understand? So if the question says that which of the following enzymes lacks the proton pumping activity, okay, but towards what? Towards the intermembrane space, because it cannot pump the protons towards the intermembrane space, right? But it has proton pumping. It can simply transmit the proton to the next complex, but not to the intermembrane space. Did you understand the difference? Did you understand the difference, what I'm trying to say? What I'm trying to say is that the only respiratory enzyme which cannot pump the protons into the intermembrane space is the complex 2. However, it can pump. If you see here, look at the diagram over here. If you see over here, it is able to pump the protons where? Okay, here, and then from here it pumps the protons to the site complex 3. That means it has proton pumping activity, but those proton pumps can be sent to the complex 3 and the complex 4, but those protons cannot be pumped over here. So this is an exception over here. So you have to remember this exception. Okay, then rest all the four complexes over here, whether we say complex 1, 3 or 4, they all can function as the proton pumping the proton into the intermembrane space. Okay, that was about the complex 2. Okay, now we move on to the complex 3. Okay, so complex 3, just remember these names. Okay, we're not going to details of this. Okay, I'm not going to tell you that we have a heme group in the cytochrome B and all that. No, 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 no. We just need to know the components over here. So complex 3, as you can see here, we have the cytochrome B over here. Okay, and then we have the cytochrome C1 complex over here. Okay, this has 11 subunits. If you remember, I told you this is the largest of all. This has 40 subunits. This enzyme has 40 subunits, but this enzyme has only 11 subunits. Okay, so we have cytochrome B over here, right? We have cytochrome C1 over here, and then we also have a coenzyme iron FeS, iron sulfur over here. Okay, the way we have iron sulfur here as well as a coenzyme. So iron sulfur is working, acts as a coenzyme over here. Are we okay about it? About the complex 3? Any questions till now? Okay. Now moving on to complex 4. Okay. And what about complex 4? Complex 4 has almost 13 subunits. So it's kind of a little bigger than the complex. Sorry, complex 4 has 13 subunits. It's bigger than the complex 3, right? So here we have, this is called as the cytochrome oxidase complex. Okay. If you see here, we have a cytochrome A here. We have a cytochrome A3 here. You will see that it's simply, this complex is very important because it will transfer the electrons at the end to the oxygen. The final acceptor of the electron is our oxygen. A very, very important point. Sometimes the question says that which is the final acceptor of the electron? So you have to say it's the oxygen is the final acceptor of the electron. When it accepts the electron, accepts the proton, it converts it into water. Okay, this is very important thing which you have to remember here. Okay, so this was about the complex B. I will tell you one good thing about complex B. Not good thing, you might have heard about cyanide poisoning. You know, you- have you heard about this? What is cyanide and what is cyanide poisoning? God forbid somebody eats the cyanide, instantly the person dies, right? The reason for is the why does a person die? Because the cyanide binds to this complex 4. Okay, cyanide, we all know that cyanide is a deadly poison, right? It straight away binds to complex 4, khalas, and that's how the person dies. Because this complex 4 is very, very important, because this complex 4 is the only component that can directly react with the O2, transmit the electrons to the O2. That is why this complex 4 is very, very important. When all the electrons will have come from here and here and here, and finally when the electrons reach over here, and it's the complex 4 which is going to give these electrons to the oxygen, right, and eventually form the water. So that is why the cyanide binds to this complex 4. So that's why a person will instantly die. Okay, this was a fact which I wanted to tell you. Okay, are we okay about the respiratory enzymes, 4 complexes? Are we okay about it? Any questions about it till now? Okay, all good. Okay, so complex 1, 3, and 4. We said that they are not only electron acceptors, they can pump the proton towards what? Intermembrane space, right? Complex 1, 3, and 4, right? We said that they not only transport electrons, but they also act as the proton pumps, right? Can you tell me that from where do they pump the proton? I know that they pump the proton into the intermembrane space, but where do they get those protons from? Complex 1, 3, and 4. From this is also the whole thing is mitochondria. This is the mitochondria. This whole thing is mitochondria. This is the outer membrane of mitochondria. This is the intermembrane space. This is the inner membrane of mitochondria. This is the matrix of the mitochondria. So the whole thing is mitochondria only. No, they send it to the intermembrane space, but where do they get those protons from? We'll discuss all that in detail, but I'm just asking you so that because you didn't ask me that question. I told you that the protons will be pumped here in the intermembrane space, but you didn't ask me from where do they get the protons from? Matrix, absolutely. Here, absolutely, from the matrix. So remember that the protons are pumped from the matrix, because in the matrix you will have NADH. Remember that your Krebs cycle took place in the matrix of the mitochondria. That is why you have NADH and FADH2 here in the matrix. Do you understand what I'm trying to say? So you have to say that the protons are pumped from the matrix towards the intermembrane space. So that is why I asked you this question. What is the intermembrane space? Intermembrane space is the space between the, this is the outer membrane of mitochondria, this is the inner membrane of mitochondria. I want to show you the, look at this. This is the outer membrane, this is the inner membrane of mitochondria. So this is the space which you're talking about. So protons will be pumped over here in the intermembrane space, this space over here. Are you able to understand? Yes, ladies, boys, anybody? Are we all okay about it? Any questions till now? Now, should we start discussing? We also spoke about, yes, so matrix to the, from matrix to the, let me get it, from matrix to the intermembrane space. Okay, now the next question, which is the final electron acceptor in the chain? Name the final electron acceptor in the electron transport chain. It's the oxygen. Okay, what does this oxygen use? These, oxygen is going to use these electrons, right, along with the protons. So oxygen is converted into, by using those protons and the electrons, oxygen is converted into the, yes, it forms the water. Very good. So we are done with all this, right? Now let's start discussing what is happening one by one. Okay, so let me erase all this first of all. Okay, so now that we have NADH and FADH2, which are produced in, which were produced in the Krebs cycle, what will happen? The electrons will be transferred from this NADH and this FADH2 to these electron transport carriers. Now you know what electron transport carriers, right? So electrons and protons will be released from this NADH, from this FADH2. So two things are released. One, electrons and protons are released from this NADH and from this FADH2. Okay. So the electrons which are released from this NADH or FADH2, they will pass from these carriers, right, from these electron carriers, right, from one to the next, then to the next, right? And as they will pass through these carriers, so the various oxidation reduction reactions will take place. Okay, from complex 1 to complex 3 to complex 4, right? What will happen at, and complex 1, 2, 3, and 4. And while doing so, the complex 1, 3, and 4, they will also function as the proton pumps. Okay, and they will move the protons from the mitochondrial matrix into the intermembrane space. Okay, is it okay till now? Okay, so when you will see that when each proton pump will accept the electron over here, right? So it will use some of its energy to, now the question is, how will these protons be pumped to the intermembrane space, right? So when each proton pump will accept the electron, okay, it will use some of the energy to export that proton into the intermembrane space. And then electron will be given to the next carrier, then next carrier, then next, and the next carrier. And finally, it will be accepted by the oxygen. Okay, so this is basically this is the basic aim what is going to happen here, right? So now in the step 1, right, as we said that in step 1, NADH, right, it has the electrons as well as it has the protons, right? So complex 1, this is my complex 1, this is my complex 1, right? It is going to oxidize this NADH, and as you can see over here, the NAD positive is produced. Okay, and this COQ will be used as an oxidizing agent. Okay, so we call this COQ as a coenzyme Q, because if you remember, I told you it can move everywhere. It's hydrophobic in nature. So when it gets attached to the complex 1, it can be used as a coenzyme by complex 1. So this NADH will be oxidized to the NADH will be oxidized to the NAD. Are we okay? So NADH will deliver its what? Its protons where? To this complex 1. Okay. And as the electrons will move, some of the energy will be used to push the protons over here. So I will have protons over here, right? Which are sent over here. Now what happens? Now there are two things happening at the same time. The protons are being pumped, and at the same time the electrons are being accepted over here, right? So I had FMN. You don't have to go into these into so into details. So is if, which one is which complex, which part of the complex one is going to accept the electron. So I'm just telling you for your knowledge, it is the FMN which is going to accept the electron. It will further give this electron to the FES. I already explained you what is FES and FMN, right? This is the flavin mononucleotide. And then finally from the FES, the electron will be given to the COQ, right? COQ is the coenzyme Q. Are we okay till now? So we pumped the protons over here, and at the same time we transmitted the electrons from the NADH to FMN to FES. From FES we transmitted it to COQ. Any questions till now? So two things happening at the same time. Protons being pumped and the electrons being, okay. Now once you have the, now what will happen to the complex 2? Complex 2, if you remember, I told you that doesn't have the proton pumping activity, but we have FADH2 over here, because FADH2 was also produced. If you remember, it was not only the NADH which was produced, FADH2 was also produced over here, right? So what will happen then? The FADH2 will transfer its hydrogen atoms, right, to the complex 2. Okay, it will, the FADH2 does not transfer its hydrogen atoms to the complex 1. You have to remember, FADH2 will transmit its hydrogen atoms, okay, to the complex 2, which is this one. Okay, now what's going to happen now? As the hydrogen atoms are delivered to the complex 2, what's going to happen here? Okay, so the electrons will move from the complex 2 to the COQ, isn't it? And then these electrons will also be given to what? To the complex 3. So we gave electrons from the NADH to the complex 1. From complex 1, we gave it to COQ. From COQ, we gave it to complex 3. Okay. And the same thing happened here. From FADH2, we gave the electrons to what? To the complex 2. From complex 2, we gave it to the COQ. From COQ, we gave it to the complex 3. Tell me how many of you did not understand this? Okay, till now we're clear. Okay, now where are we right now? We are right now. Okay, so we are right now where we have FADH2 came from the Krebs cycle. Yes, Mohammed, right? That is why, see, I'm going step by step. That is why I told you that FADH2 and NH2DH was the product of the Krebs cycle. Okay. Yes, Mohammed. Clear? Okay. Now what happens, as I told you that here now, okay, we are at the complex 2 right now here, right? So what happens when this FADH2, okay, will be oxidized to the FAD, okay, will send the hydrogen atoms over here or send the electrons over here. What happened? The energy produced over here is, is not sufficient enough to pump the protons across the membrane. And if you remember, nor we have a channel over here which can pump the protons into the intermembrane space. Okay, so no protons are pumped at the complex 2. This is a very, very important step, or you can say that complex 2 lacks the proton pumping activity. Okay, now after that we reach to the complex 3, right? What's going to happen in the complex 3? Again, the same acceptor of the electrons. If you remember, I told you that it has plenty of things which can accept the electron over here, right? If you see, we said that it has cytochrome C1, then it has cytochrome B, right? So electrons will be accepted by these components in the what? In the complex 3, right? So complex from COQ, now the electrons are where? The electrons are now transferred into the complex 3, right? So from the complex 3, now the electrons will be transmitted to the cytochrome C. Okay, so what is happening throughout this when the electrons are being transferred from one complex to the other complex. So every time each complex is first getting reduced, isn't it? Okay, and then as it transmits the electron, then it's getting oxidized. Why reduced? Because it is accepting an electron, okay, and then passing the electron to the next one, okay, and then it's getting oxidized itself. So it's accepting the electron, getting reduced, passing it to the next complex, and then that will get reduced, and itself it will be oxidized. So that is what will happen till from cytochrome C, the electron will be given to the final complex, which is the complex 4. Okay, and as if you remember, I told you this complex 4 is very, very important because it is this complex 4 which will finally give the electrons to the oxygen and produce the water, right? So while the electrons are being jumped from one electron transport chain to the other electron transport chain, from the complex 2 also we will have protons present over here, not outside. I'm sorry, I'm going to delete it. Give me a minute. So if you see here, we will have protons here. Okay, we'll have protons here. From complex 2 also we will have protons pumped over here. We'll have protons pumped over here, and complex 4 also will have protons pumped over here, right? Okay, so bumping of these protons produces a concentration gradient over here, right? So higher concentration of the protons will be over here, right? And now then now comes the final step of this, how now to generate the ATP. But before I can start describing how do we generate the ATP, did you all understand what is happening in the at the complex 4? At the complex 4, the electrons which were accepted by complex 4, right, which originally came from NADH or came from FADH2, right? So oxygen here has a greatest affinity towards these electrons, right? So two protons and the electrons will combine with the oxygen. In simple sense, you can say that oxygen pulls the electrons. It has such a great affinity towards the electrons, it will pull the electrons, right? And you will see that the water will be produced here. Okay, and if you see here, I've made these steps here to show you how electrons jump from one electron carrier to the next, right, till it reaches to the end where the formation of water will take place. So with that, we finished one part of it, which is the electron transport chain, right? And then we have the next part about it, which is the how the ATPs are synthesized by a special type of enzyme, which is this one, the ATPA synthetase. So should I explain about that now? Any questions till now? Okay, now the last part of this chapter. Okay, so you will read all this. So it has the same thing what I explained it to you. Okay, one getting oxidized, another one getting reduced. Okay, so usually we have 6 protons are pumped from the NADH2 and from the NADH, and 4 protons are pumped as a result of FADH2, okay, into this intermembrane space. Now the last part of it is the how exactly do we generate the ATPs. Okay, in order to understand that, first of all you need to know the structure of the enzyme. Okay, and in order to know the structure of the enzyme, you also need to know what is the chemiosmotic theory. Okay, so I'll again go to the diagram over here so that I can explain you what is the chemiosmotic theory and how ATPs are produced. Okay, I'm going to explain you first of all about this special enzyme which is called as the proton translocating enzyme, or we also call this enzyme this. Okay, we also call it an ATPase synthetase enzyme. We also call it as an F0 F1 enzyme, and I'll tell you why it's called an F0 F1 enzyme. Okay, or simply we call it sometimes the proton translocating enzyme. Okay, so this is the enzyme which we'll talk about now, because you will see that is this enzyme which will help in the formation of the ATP. Okay, now if you see this ATPase enzyme, it has one subunit which is called as an F0 subunit. Okay, if you look at this F0 subunit, this subunit is present within the inner mitochondrial membrane. This is my inner mitochondrial membrane, right? And it is F0 is present within the membrane. Okay, now what happens, it also has an extra membrane domain. Okay, this enzyme also has a part of it which is not present within the inner membrane, which goes outside it. Do you see it here? This one, this one. Okay, so this is called as an extra membrane domain, or this is called as an F1 domain, or we sometimes call this as the headpiece. This is also called as the headpiece. So I said that we have a special enzyme called as an ATPase synthetase. Okay, that enzyme has a portion which lies within the membrane, which is the F0 membrane portion. Okay, this one. And then it also has a portion which goes out of the membrane, which is an extra membrane domain, which is called as an F1. So that is why ATPase synthetase enzyme is also sometimes called as an F0, F1. Okay, now since now we know, and then this F1 has different subunits as an alpha, beta, beta. You know what are the subunits now, right? After knowing the quaternary structure of the proteins, you know what are the alpha, what are the subunits now, right? We don't need to explain you about what are the alpha, beta, right? They're all the subunits of this enzyme or this protein. Okay, so we had a scientist. Okay, he told us about the chemiosmotic theory. Okay, what is the chemiosmotic theory? Okay, he told us that when you have, now listen everyone carefully, let me erase first of all this. Now we know that we have a lot of protons over here, right? A lot of protons over here. Okay, see, he told us that in the chemiosmotic theory says that, okay, whenever there is a proton gradient created, okay, on the intermembrane side, okay, what will happen that these protons, okay, they will re-enter the matrix. They will so protons which are present in plenty in the intermembrane space, as per this theory, these protons are going to re-enter. As you can see here, this is the channel through which the protons can re-enter. Where? Okay, where will they re-enter? From the intermembrane space, the protons will re-enter. Let me see in the matrix, absolutely right. And they enter through a special channel protein, right? And that channel protein is present in the F0 subunit of the enzyme called as the ATPA synthetase. Okay, and please pay attention now. As these protons, when they travel through this channel, okay, what happens when they move into the matrix? What do happen? What will happen? The influx of these protons, okay, they will drive this rotor, okay, or the components of the F0. You can say that the F0 is rotated. This F0 is rotated. Okay, as the protons travel over here, the F0 is, as you can see here, it rotates. There will be a conformational change. Okay, in this F0, the conformational change in this F0 is further going to bring the conformational change in three subunits of F1. Okay, what are those three subunits? This beta, and this beta, right? So rotation of the F0 will cause a conformational change in the F0 component, which will further change what, or rotate this, or produce a change in the beta subunit of the F1. As the beta subunit of the F1 is rotated, or you can say they change their conformation, it allows this beta subunit to bind to the ADP, right? So ADP gets phosphorylated. That is what it needs. ADP has to be phosphorylated. So ADP gets phosphorylated to form the ATP. So that is how ATP is synthesized in the F1 portion of the ATPase enzyme. Sometimes the question says that ATP is produced on dash part of the ATPase synthetase enzyme. So remember, it is the F1 part of the ATPase synthetase enzyme. So thus we can say that this enzyme, the ATPase synthetase enzyme, what happens? This enzyme uses the energy which was released from the protons, which moved down their concentration gradient. They were more in number here as compared to the matrix. So as they went to the down their concentration gradient, what happened, right? As they moved down their concentration gradient, in this process, the ATP was synthesized from the ADP, because ADP was phosphorylated by the beta subunit of the F0 enzyme. Okay, so I will not go into details. Basically, there are other concepts also involved here. It actually generates a proton motive force. Okay, so we usually say that the protons which are present over here, okay, they have a potential energy. I will not go into all those details. You don't need to know all those details. So that potential energy basically is called as a proton driving force. So it's that proton driving force which is basically catalyzing the synthesis of ATP from the ADP. But you don't need to know all that. You simply need to know how the ATP is produced, right? So in short, the energy stored in these protons, okay, they will drive this component. They will make the conformational change in the F0, which will further make the conformational change in the beta subunit of the F1, and which will which will cause a phosphorylation of the ADP, and that's how the ATP is produced. With that, we finished the last part of the what? Of the oxidative phosphorylation. Is everybody tired or nobody's asking any question? Did you all understand everything? Are you superficially saying yes or you really understood it? You really did, right? Really understood it. Okay, you have to be honest. So, okay, let me ask you a question. Now, what did we generate in the Krebs cycle? What was the importance of Krebs cycle? Anybody can, you can switch on your mic if you want to. NADH and FADH2. Wonderful. Very good. Wonderful. Everybody's replying. Okay. So now we have NADH and FADH2. So how did we use this NADH2 and NADH and FADH2 in the electron transport chain? They do not only very good. They not only donated the protons, they also gave the electrons. Oh my God, you guys are amazing. I thought you really did not understand. Oh, everybody is replying. Very good, Alhamdulillah. They gave electrons and they gave the protons as well, right? So not only remember that not only electrons, but the protons are also released from the NADH and the FADH2. Very good. Then what happened? While electrons were transmitting from one complex to the other complex, what happened? When they were transmitting from, yes, some of the energy was released. And what did we use that energy? Very good. Oh my God, everybody is replying. Yes, we used that energy to pump the protons. So where did we pump the protons? From where to where? Very good. We use that protons to pump from, tell me, from and to as well. Yes, very important. From matrix to the intermembrane space. Wonderful. Very good. Everybody is replying. Okay. So protons, yes. So protons were pumped from the matrix into the intermembrane space. Wonderful. Okay. So as a result of that, what did we have in the intermembrane space? What did we have in the intermembrane space? We got a proton accumulation of protons. Wonderful. We got a proton gradient. So that means I had more protons on the intermembrane space as compared to the matrix. Okay. Okay. Let's finish with the protons first. Then we'll come to the electrons. So what did we do with those protons? So how did this proton gradient help us? All the girls are writing. I didn't see any boy writing answer. Boys, where are you? What happens to that proton gradient once we have the proton gradient? Protons can be sent back to the matrix. Very good. Wonderful, Lean. Yes. Protons can be back to the, yes. So as you send these protons back to the matrix, what happens? Let me erase all this. As you send these protons back to the matrix, eventually what do we do? In between, what do we have? We have an enzyme by the ATP synthetase. Yes. So those protons are used to rotate F0 component. Very good, Tanya. When the F0 component rotates, it changes its conformation. It further changes the conformation of which subunit of the F1? Beta subunit. Wonderful. Oh my God, you guys are amazing. Alhamdulillah. Alhamdulillah. This is like the most difficult concept of, respiration, wherein students, they don't understand why we are learning all this. Okay, great. So once we have the beta component changing its conformation, eventually it phosphorylates what? It phosphorylates the ADP to form the ATP, and this process is called as the chemiosmosis. This formation of ATP from the ADP using the ATP synthetase by using the energy of the protons, we call this as the chemiosmosis. Osmosis. Wonderful. You guys are amazing. Okay. Then what happened to the electrons? We didn't speak about electrons. Electrons, as they jumped from one complex to the other electron carrier to the complex 3, then cytochrome C, then complex 4, then what happened to them at the end? From cytochrome A3, once we have the electrons are with the cytochrome A3, they were transmitted to the, they were picked up by the oxygen. Wonderful, right? Oxygen pulls the electron. Razan used the word which I used, very good. Oxygen will pull the electrons, right? And what will oxygen be converted into? Water, right? So now do you understand why do we need oxygen? Why do we breathe in oxygen? Why do we say it's an aerobic respiration? Do you understand now how important this oxygen is? If there's no oxygen, there'll be no electron acceptor for us. No electron acceptor, that means khalas, you cannot oxidize the food which we eat. So this shows us how important is the oxygen which we take from the by using our respiratory system. See, I didn't say anything. You all said everything. I was just acknowledging the fact. So with that, we finished the chapter. I don't think we have anything left out at this. Oh yeah, yes, 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 yes. I have one important thing to discuss over here. Yes. So with that, we finished it. Yes. Okay. Since the class time is also over, okay, and okay, okay, I will come back to this in a while. Okay, let me finish this chapter. We just have one slide left. Okay. So here it's trying to say you, trying to explain that the chemical energy present in the ATP will be converted to several other forms of the energy. For example, you saw that whether we talk about, the depolarizations in the neurons, okay, or in the muscles, okay, we use these ATPs. If you remember, we discussed that the amount of potassium should be present more inside, isn't it? While as the amount of sodium should be more outside, okay, when the cell is maintaining a resting membrane potential. We discussed all that in muscular system and the nervous system. We're not going to repeat all that, right? And if you remember, we discussed about the sodium potassium pump there. Remember the active transport? Yes, that active transport needs ATP, right? So how did we generate those ATP? Those ATPs were generated by this process over here, right? So just trying to explain you that the chemical energy of the ATP will be transformed into the electrical energy in your neurons as well as in the sarcolemma of the muscles. Also, this ATP will also there will also will help in the association and the dissociation of what? Actin and the myosin. This is what we learned in the last semester, right? The sliding filament theory, right? And eventually your muscles will contract and relax. Okay, and some amount of ATP can also be used to convert what? To release it as the heat, which will help us in maintaining the body temperature, right? So now where in the mitochondria is the concentration of H positive highest? It is in the intermembrane space. Okay, I'm just kind of wrapping up everything. In cellular respiration, the final electron acceptor is the oxygen, which we already spoke about. The electron transport chain transfers the electrons to oxygen, which is reduced to the water. Okay, as electrons travel through the electron transport chain, protons are transferred from the mitochondrial matrix to the intermembrane space. We spoke about all these things. And then oxidative phosphorylation collectively refers to the electron transport chain and the chemiosmosis. Together, it's called an oxidative phosphorylation. The only thing which I did not discuss here was about the very important part of the chapter, which was about what was the net gain of the energy. Very, very important part of the chapter. Okay. But this part of the chapter, I'm going to assign all of you as a homework. Is everybody listening to me? So I want all of you, you can either see all you need to do is, because this is such an important part of the chapter, because you are being asked at the end, what is the net gain and the net loss of energy? How many ATPs did we produce? Right? So this is a very important part of the chapter. So, but I will explain this part next week in the in Monday's class. But before that, this I'm going to assign it as the homework to everyone. Okay, so you, what you can do as a homework, you can tell me how many ATPs were produced at the end, what was the net reaction which happened, okay, after we had a Krebs cycle, okay, and after we had, oxidative phosphorylation, what was the net gain of ATPs, okay? So you can either email me, okay, you can, I would prefer to email me, because I'll tell you why I would prefer to email you, because I'm going to record everybody's response in my attendance sheet. Okay. So as you will do this activity, you will eventually read about all this, and then only you will be able to write it down or jot it down how many ATPs are produced, right? Since we're not going to have a class on Wednesday, so you can do this activity over the weekend on Wednesday, and next week, on by next Monday, you all should send me your response by email, and I'm going to record that and add that, in your attendance bonus. Yes, I will repeat once again. My question is that you all will tell me what happens, how many ATPs are produced at the end. How many ATPs did we produce in the Krebs cycle? How many ATPs we produced in the oxidative phosphorylation? And then by combining the two process, how many ATPs were there in the overall reaction? Okay, and please remember that I really want to tell to all of you is that there is something called as an old method and the new method in the Google also. Mohammed, did you send it to me through old method or the new method? One NADH is equivalent to how many ATPs? Is it the old method or the new method? Okay, so you will write it whether you did it through the old method, because both of them are different. Okay, so it is your job to find out what is the old method saying and what is the new method saying, because we have a different, we will get a different ATP count as per the new method, and we will get a different ATP count as per the old method, right? So it's your job to find out one NADH is equivalent to how many ATPs, and one FADH2 is equivalent to how many ATPs as per the old method, as per the new method. So doesn't matter, even if you send me by the old method, it will be okay with me. If you send it to me by the new method, it will be okay with me. But I want all of you to find out, to learn, to read about it, and eventually, you will prepare this topic. Okay, is it okay? Did everyone understand the question? Do you want me to post the question in Moodle? If you want me to post the question in the Moodle, okay, I will do that, right?